Okay, Hammy here, back with part 2 of section 7.1, in which we will look at the discovery of the DNA molecule and how it was sort of figured out that it was the genetic material. Now, scientists had known about nucleic acids, DNA, and RNA for a while in the early 1900s, but they did not know it was the genetic material. So I guess, after the rediscoveries of genetics, scientists became more curious as to what exactly is happening. And in the last video, you remember we talked about Frederick Griffith's experiment with the mice and how something was transforming the rough strain into the smooth strain. So Oswald Avery and a bunch of scientists that were helping him, grad students and other professors and so forth, in 1944, they were running a bunch of tests to see what was the transforming substance that was causing the R strain to turn into the S strain. So what they did is they took heat-killed S strain. Okay, so over here in the vial over here, they took the S strain bacteria, heat-killed it, filtered it, and then they treated it with different enzymes to knock out certain substances to see if it would still transform the R strain. Okay, so in your notes, the first one, the Polysaccharide Ace, okay, over here they just have that as carboase. Okay, in other words, taking out polysaccharides or carbs, they treated the solution with enzymes that would destroy the sugars. And if you look what happened, okay, after they added the enzyme to the S strain filtrate and then added it to, this is the R strain beaker right here, okay. Then when they looked at it later, it had S and R strains. So something, it was not the sugars that were causing the S strain. So there was still transformation you could write down in your notes. The next one, proteases up here, okay, proteins. When they treated that, that heat-killed S strain with enzymes that would destroy proteins, look what happened right here. Still transformation. So write down still transformed. Okay. Next, I didn't put it in your notes, but they used lipase to take out the lipids. Still transformed. Used RNase, which took out ribonucleic acid. Still transformed. Okay. Right here, this red circle, when they treated it with DNase, an enzyme that took out or destroyed deoxyribonucleic acid, okay, there was no more transformation. So, prevented transformation. Okay, and this was a key finding here, that when you took out the DNA or destroyed the DNA, the transformation from Griffith's experiment with the bacteria did not work. Okay, this led Oswald Avery to write papers and make the conclusion that that transforming substance was deoxyribonucleic acid, okay, DNA. Now, in their paper, they didn't suggest that DNA was the sort of the genetic material, although they had their suspicions. This is courtesy of the Rockefeller Archive Center here, a kind of a copy of a letter he wrote to his brother, in which he kind of made those suggestions like, hey, if DNA is causing this transformation to happen, then maybe it's the genetic material, because whatever's changing the R strain into the S strain had to have some genetic material in it. But he never publicly sort of announced this, although he had his suspicions. That debate was finally settled with Hershey and Chase. Alfred Hershey and Martha Chase, you can see their picture over here. They worked with viruses that infected bacteria. These little viruses that we call a bacteriophage. Okay, a bacteriophage has a DNA core in the middle and then a protein coat kind of all around it. They look like these little alien things almost. And how these bacteriophages work is that up here, starting up here, okay, they attach to a bacterium and then they put something into the bacteria, took over the bacteria's machinery, made basically made more proteins or more viruses. Okay, and then the viruses would be put together, and then the bacteria would explode open, and now the viruses would be released up here, and they would go infect other bacteria. Okay, called the lytic cycle. So it's so they figured out, well, if we could figure out what is going into the bacterium to take over, that's probably the genetic material. So what they did is they devised an experiment in which they used radioactivity, okay, two different radioactive elements, P32 and S35. Now, phosphorus is only found in DNA and not in protein, whereas sulfur is found in protein and not DNA. So they could label these two things and know that either the outside coat was labeled or the inner core was labeled. And when they ran their experiment, look what they found. Okay, this bacteriophages in a medium with radioactive phosphorus, and then they let the viruses infect the E. coli. They used a blender to kind of shake them loose, and this shook off the protein coat. Okay, you see this thing going flying off. And then they centrifuged it. Okay, they spun them real fast, and the heavier contents sunk to the bottom, which would be like the pellet. And then all the liquidy part at the top was liquid, and these kind of the virus part that was shook off. So all the bacteria went into the pellet, and all of the fluids and these little virus coats here went into the solution. So they tested the solution. Where was the radioactivity? When they did the radioactive DNA, the radioactivity was in the pellet, okay, which means it was in the bacteria. When they used a radioactive protein, okay, and they took the blender, removed the viruses from the bacteria that were infecting the bacteria, and then they centrifuged it, they noticed that the 35S was in the supernatant, or the liquidy part. Okay, so if you have a test tube, kind of illustrate here. Okay, if you have a test tube, you've got some liquid at the top, and then the solid part, when you spin it real fast, the solid part will all fall to the bottom in kind of this little pellet. And then you'll have this like liquidy stuff up here. Okay, that was a supernatant. So when they used radioactive phosphorus, okay, they found it in the pellet. Okay, after they centrifuged it. Okay, and then which was where the bacteria were. When they used the radioactive sulfur, okay, they found it in a supernatant, or the liquid part, okay, which would not be in the bacteria. So if phosphorus is only found in DNA, and that's what was found in the pellet where the bacteria were, this kind of finally solved it, that what is the genetic material, what is taking over the host bacterial cell. They were the ones that kind of concluded that genetic material was in fact deoxyribonucleic acid. And then they published papers and kind of publicly announced this, and this sort of then began the race to figure out, everyone agreed that DNA was a genetic material, and then the race began to figure out, how does this DNA stuff work? How does DNA actually store our genetic code? And that's the topic of our next video.